418.3. Yokota Air Base quiet hours in effect. Control tower is closed. Pilot-operated approach lighting is not available. For weather and additional information, contact Yokota approach on 270.64118.3. Yokota Air Base quiet hours in effect. Control tower is closed. Pilot-operated approach lighting is not available. For weather and additional information, contact Yokota approach on 270.64118.3. Yokota Air Base quiet hours in effect. Control tower is closed. Pilot-operated approach lighting is not available. For weather and additional information, contact Yokota approach on 270.64118.3. Yokota Air Base quiet hours in effect. Control tower is closed. Pilot-operated approach lighting is not available. For weather and additional information, contact Yokota approach on 270.64118.3. Yokota Air Base quiet hours in effect. Control tower is closed. Pilot-operated approach lighting is not available. For weather and additional information, contact Yokota approach on 270.64118.3. Yokota Air Base quiet hours in effect. Control tower is closed. Pilot-operated approach lighting is not available. For weather and additional information, contact Yokota approach on 270.64118.3. Yokota Air Base quiet hours in effect. Control tower is closed. Pilot-operated approach lighting is not available. For weather and additional information, contact Yokota approach on 270.64118.3. Yokota Air Base quiet hours in effect. Control tower is closed. Pilot-operated approach lighting is not available. For weather and additional information, contact Yokota approach on 270.64118.3. Yokota Air Base quiet hours in effect. Control tower is closed. Pilot-operated approach lighting is not available. For weather and additional information, contact Yokota approach on 270.64118.3. Pilot-operated approach lighting is not available. For weather and additional information, contact Yokota approach.
Good afternoon. Happy Friday, everyone. Happy Friday. 1:09 p.m. Eastern Standard Time on Friday afternoon, October 6th. That means I need to pay some bill today. October 6th, year 2023. My name is Mark Kulak. This is our 245 series where we are reading an article, a multi-section article called The Story of Cesar Milstein, and the story of antibodies, monoclonal antibodies. This is part 4, and we are on our fourth chapter, fourth section, if you will. The previous 3 sections or readings I broadcasted on our backup channel, Houstonic Live 2. We chose Houstonic Live 2. Some of this stuff can be a little spicy, especially when we add in a little extra narration. So this is one of the longer sections out there. And this section is called Making Monoclonal Antibodies. By the way, for those looking for this article, what is biotechnology.org? What is biotechnology.org? No spaces, no hyphens. What is biotechnology.org? You can go there. You can find this. I should have a link to it. The article's date is about the year 2012, approximately. So, with that said, let us make sure things are streaming here. Are they streaming at all? I think that they are streaming right now. I see 11 watching. That's excellent. All right. We're just going to dive right into it. This is a longer article. Section anyways. This section is Making Monoclonal Antibodies. The first part, the hunt for a single antibody. From the late 1960s onward, Cesar Milstein would devote much of his research to understanding somatic mutation in antibodies. The idea that somatic mutation could help explain antibody diversity was not new. Instead, Milstein and Sidney Brenner's suggestion that this could be what determined antibody diversity was in many ways an extension of a hypothesis first formulated by the American molecular biologist Joshua Lederberg in 1959. He is one of the most interesting people you will read about, among the most anyways. Joshua Lederberg, biotechnology, biodefense advisor to at least 7 United States presidents, close, long-time friendship, relationship with Henry Kissinger. He passed in the, I think, 2007, 2008. He won a Nobel Prize in his 30s for work that was done on, effectively, bacteria replication. And it was research that Joshua Lederberg did where he realized that bacteria just were not splitting and dividing endlessly. That bacteria actually had ways of reproducing that involved the exchange of genetic material between parent, if you will, bacteria. You may want to call it sexual reproduction. It's sort of akin to that. Not all, but there are many types of bacteria that actually exchange genetic information before having offspring or reproducing. Isn't that amazing? And what is even more amazing about that statement is that with bacteria being as small as they are, they actually use viruses to do that. They use, these are bacteriophages, of course, being bacteria, which is what he was studying. But what Joshua Lederberg bumped into was a realization that there are virus-like particles which appear to be playing a role which wasn't just invading cells and killing things. It was a role that was vital to the survival of multiple different types, multiple strains of bacteria. That statement is amazing. And it was actually the work of Lederberg that has had greatly impacted my own views of viruses. And again, to say that viruses do not exist, one must explain, well, how is it that so much bacteria manages to then reproduce using viruses? You need to acknowledge that some type of molecular biology exists and is real. Without acknowledgement of that, there's just too much is being denied. Let's see here. We have, I apologize. I'll make sure. Wow, only 11 people here. That's excellent. You guys are amongst good company. So we've done extensive work researching Joshua Lederberg. I think he has provided one of the most important insights. It has opened up the doors to revisiting what the heck viruses really are, and has led to our assertion that viruses, whatever they are, whatever they are not, they exist only within the realm as a subset of molecular biology. And whether or not you want to categorize them as viruses or not is purely it's a language game. Hey, Gene, how are you? Anyways, I have a lot of shows on him as well, all the way up. He even had roles in September 11th with respect to the research after the events of September 11th, working directly with Rumsfeld. I mean, he's just such an impactful character. So the photograph that we see here, and I'm going to get a little bit smaller on this screen here so that we can share this photograph. This photograph shows myeloma cells stained purple as they appear under a microscope. So these myeloma cells, these are full cells. So obviously, I don't believe the nature at this scale has such beautiful, vibrant colors. So much of what we see, like when Vincent Ronquiniello has all of these, and the Debunk the Funk Bob Wilson, have all of these beautiful color, vibrant, shiny virus particles on their shelves and hanging and spinning like they're little globes at a discotheque. That's all imagination. Those types of colors don't really exist. That's actually their passion and appeal for viruses has colored them in. So when you do see these beautiful photographs, because of the technology that's used to photograph these things, color is just different at that scale. Anyways, so in his quest, in Cesar Milstein's quest to understand somatic mutation and antibody diversity, Milstein was greatly helped by the availability of cells taken from the blood of patients with multiple myeloma. So before we go forward, I did want to just pull up the Wikipedia definition of what somatic mutation is. It's a change in the DNA sequence of a somatic cell of a multicellular organism with dedicated reproductive cells. That is, any mutation that occurs in a cell, that is a germ cell, a gametocyte, etc. Unlike germline mutations, which can be passed on to descendants of an organism, somatic mutations are not usually transmitted to descendants. So, when two beautiful creatures of God decide to have a child, somatic mutations are generally not passed on, as this diagram here shows, right? Somatic mutations occur earlier in embryonic development, are generally present in a larger fraction of the body. If, as in this case here, the mutation happened in early development, then in G, therefore in present. Okay, so there's some type of mutations, I guess, which can get passed on. Child has a mosaic somatic mutation that occurs early and post zygotic development and is present in a percentage of his cells. So there's some types of mutations which very, very early on, possibly even impacting the egg or the sperm, can be passed on, but many of these other types of mutations are not. Okay, so just wanted to have a little bit of a background there. So in his quest to understand somatic mutation and antibody diversity, Cesar Milstein was greatly helped by the availability of cells taken from the blood of patients with multiple myeloma. Such myeloma cells, which produce antibodies resembling normal antibodies, had been noted in 1951 by Henry Kunkel, Kunkel, an American immunologist based at the Rockefeller Institute in New York. Investigating the blood of myeloma patients, Henry Kunkel was surprised to observe that malignant plasma cells of multiple myeloma appeared to produce just one antibody. This contrasted with normal plasma cells, which produce a vast array of antibodies. This is interesting. So the cells themselves are producing the antibodies. That's interesting. Or just one type of antibody. Henry Kunkel's discovery was a significant breakthrough for antibody research. Ever since the early 20th century, scientists had been struggling to isolate and purify single antibodies from the billions made by the body every day. One of the advantages of the antibodies produced by myeloma cells is that they are all identical. Moreover, they are fairly easy to obtain in large quantities from the blood or urine of patients with multiple myeloma. Based on this, Henry Kunkel and his coworkers began using myeloma cells as a tool for investigating normal antibodies, because it's a predictable baseline. The availability of myeloma cells for antibody research greatly expanded after 1962, when the American molecular biologist Michael Potter, I wonder if any relationship there, Michael Potter, Potter, made the serendipitous discovery that an injection of mineral oil into the peritoneal cavity of BALB/C mice. I don't know what that is, which is a particular strain of laboratory mice, induced the growth of myeloma cells. This meant that the cells could be grown easily and indefinitely. Following this, Potter and his colleagues at the National Cancer Institute in Bethesda accomplished a collection of myeloma cell lines for distribution to researchers around the world. This is so the availability of myeloma cells for antibody research after 1962. American molecular biologist Michael Potter. Now that year, 1962, really sticks out here. Michael Potter. That's definitely not him. Immunologist. He worked with Paul Ehrlich and won the Paul Ehrlich Ludwig Darmstädter Prize. Monoclonal antibodies. He attended Princeton. Had 2 children. Interesting. I wonder what year that big discovery was, though. And I say that because of Henrietta Lacks. And sorry to drift from here. So Henrietta died 1951. Okay, thank you. I was wondering. I might have had that date wrong. Okay, so she passed in 1951. Because I noticed that in 1951, 1952, a whole lot of scientists seem to have suddenly made big jumps in their ability to get cells to reproduce. And we are speculating that was the Henrietta Lacks story, in some cases anyways, used as a cover for doing cell reproduction in other ways, to say, oh, we got it to work thanks to Henrietta, the HeLa cells. But in reality, while no one was looking, maybe fetal cells were being swapped in. Who knows? We are free to speculate about stuff like that. So the availability of multiple myeloma cells for antibody research greatly expanded after 1962, when the American molecular biologist Michael Potter made the serendipitous discovery that an injection of mineral oil into the peritoneal cavity of BALB mice, a particular strain of laboratory mice, induced the growth of myeloma cells. Wonder why it made him do that. This meant that the cells could be grown easily and indefinitely. Following this, Potter and his colleagues at the National Cancer Institute in Bethesda established a collection of myeloma cell lines for distribution to researchers around the world. Interesting. So the mice, it's a particular strain of laboratory mice, but all these doing is injecting mineral oil. So I just wondered how humanized these antibodies are. They have to be some type of. They can't possibly just be mouse antibodies, right? By the 1970s, scientists' access to myeloma cells was made much easier by the work of Kengo Horibata, Horibata, Kengo Horibata, and A.W. Harris, under the supervision of Melvin Cohn, Cohn, at the Salk Institute in San Diego. Woohoo! They found a way of adapting Potter's mouse myeloma cells to grow in tissue culture, a technique that allows for the growth of tissues or cells outside of an organism. Essentially, tissue culture provides for the growth of cells under controlled conditions outside their natural environment. The growth of such cells is helped by the use of a suitable medium, which provides the nutritional elements they require. In this world of cell culture, it's my understanding is this is an art. There is the people who can keep the cell lines alive and reproducing and going from generation to the next. This is an art. Things happen that they don't understand, and they just put it in the books. They put it in the notes. As someone once explained to me, if you're trying to get a particular group of cells to reproduce, and one day you accidentally knock it off the table, and someone walks by with a shoe where they were just at the gym, and then you scoop it back up into the dish, and then it starts to grow again, well, then guess what? That becomes part of the routine. That means every time you want to grow the cells, you got to take it on a table, knock it onto the floor, find a shoe that was just at the gym, stomp on it, put it back into the dish, and then put it back onto the table. You just start repeating things that work, even though you have no idea why they were actually part of the process in the first place. Also, why, for those who are researching the history of what's described as cancer-causing viruses, which is really probably cancer-causing exosomal research, but either way, look at the SVCP, the Special Virus Cancer Program section on who managed cell cultures, because those are among the most important people. When those people go away, the cell lines go away. The cell culture people were among the most important people in the Special Virus Cancer Program. When you're looking at older research papers, look at the notes at the end, the small font, or the little subtext, the little appendices, where they'll say, guess what? This person provided this cell line to this person. Sure enough, you will find papers around 1950, 1951, 1952 regarding Hillary Koprowski, who is in this article, by the way. And his cell cultures were provided to him, at least in that case, by Alice E. Moore, the doctor of the Memorial Sloan Kettering Hospital. You have so much catching up to do. We all do, Kim. We all do. I'm glad you can still feel that way. As again, we go through our life adjustment here. I'm just, it's good because we want to maintain the growth. I'm not looking to take a break from that. But it's good. I'm kind of pleased that you actually can feel that way. That's good. At the end of this reading, by the way, not just probably today, I'll probably take a few minutes before I end this episode. I'm going to go back. I'm going to make a list of all the names. There's a lot of names in here. I mean, I've already just encountered 5 names that I've never even seen before myself. It's bananas. The story of, as I mentioned in the first episode, why we're doing this. I thought antibodies would be boring. I did not think antibodies, I thought the real action was going to be studying the viruses. I thought the real action was going to be the geneticists. But it's amazing how this article, not this section here, but even yesterday's article, clearly said, and I will go back to what I read yesterday, because I think it really is that important. It's, and then I'll reread the paragraph of where we just left off. Okay, this was from part 3 yesterday. By the early 1970s, Cesar Milstein had begun to shift his research towards investigating the role played by messenger ribonucleic acid, mRNA, and the encoding DNA in determining the diversity of antibodies. The mRNA is a type of chemical blueprint for a protein product. I'll read it one more time. By the early 1970s, Cesar Milstein began to shift his research towards investigating the role played by messenger ribonucleic acid, mRNA, and the encoding DNA in determining the diversity of antibodies. The mRNA is a type of chemical blueprint for a protein product. In other words, they were studying mRNA not because they thought mRNA itself was that awesome. They were studying mRNA to learn about antibodies. They wanted to learn more. They wanted to get more intellectual property about antibodies, and they were hitting a wall, and they found the study of mRNA to be useful for that. you'd think, and I've been brainwashed into thinking, oh, the antibodies are just something that you measure, and all the action, all the juice is at the mRNA level. And this flips that around. And that is why, so far, this article, you wouldn't think it. It has Francis and Crick and Lederberg and Sidney Brenner and George Pachenik, brother of Steve Pachenik, and Hillary Kaprowski. They're all here. Everyone. The virus cancer program, everything is going to be in this article about antibodies. Because what we thought at times was just the study of exosomes, viruses, whatever you want, it will. Rafting, as a fun phrase used by Sina Bavari, they're all really studying protein expression. Antibodies are proteins. Protein expression. What is nanotechnology? Nanotechnology is the new machinery at the molecular level. Nanotechnology is proteins. Proteins are nanotechnology. Not little tiny computers, not tiny Intel chips, not microscopic versions of computers or little pistons or tires or wings or whatever. Yes, I know we can make very small little, planes which can do things, maybe like make them as small as a bottle cap or something like that. Yes, that technology definitely does have military applications, but nanotechnology is molecular biology. And at the molecular level, the way machines work is completely different. The nanotechnology initiative is this, and everything in nanotechnology is a protein. And proteins are made by ribosomes. So this whole initiative and all of these efforts involve proteins and the nanotechnology initiative and ribosomes and so on. Anyways, so you'd think, oh, mRNA, that's the future. It's like, well, it was being pushed. And as in that 3-minute clip I showed from Drew Weissman, that 3-minute clip, the Nobel Prize winner, he was reflecting back on an interview where someone asked him about mRNA several years ago, 10, 20 years ago. And even he, the Nobel Prize winner, said that he did not think at that time there was any useful thing for any useful application for mRNA. That was amazing. And he then went on to confess that the recent bonanza investment in mRNA, I think we have some trees being cut down at a neighbor's house, was about improving efficiency of making antibodies. But anyways, you have to watch the 3-minute clip. So I want to go back. This reading may take a while because obviously I'm energized and I'm feeling the need to do a lot of little tangents here. So if you don't mind, we're going to have to go back a few paragraphs and reread this yet again. I'm going to go back 3 paragraphs, as painful as that may be. In his quest to understand somatic mutation and antibody diversity, Cesar Milstein was greatly helped by the availability of cells taken from the blood of patients with multiple myeloma. Such myeloma cells, which produce antibodies resembling normal antibodies, had been noted in 1951 by Henry Kunkel, an American immunologist based at the Rockefeller Institute in New York. Investigating the blood of myeloma patients, Kunkel was surprised to observe that malignant plasma cells of multiple myeloma appeared to produce just one antibody. This contrasted to normal plasma cells, which produce a vast array of antibodies. So they found one. So different cells can usually produce each a vast array of antibody types. So these weird cells, these cells from patients with multiple myeloma were only producing one antibody type, right? It's kind of like, you want to find, I don't know, I'm trying to come up with a stupid example here. You have your general purpose chef, and then when it comes to the multiple myeloma cells, they are only capable of making grilled cheese sandwiches, and that's it. The same type of cheese, the same type of bread. No variation whatsoever. That they can make one thing when it comes to antibodies. I don't know why, but that observation was helpful when they wanted to study antibodies. They wanted to at least narrow down somewhere they can get just one type of antibody. So this Henry Kunkel's discovery was a significant breakthrough for antibody research. Ever since the early 20th century, scientists had been struggling to isolate and purify single antibodies from the billions made by the body every day. One of the advantages of the antibodies produced by myeloma cells is that they are all identical. Moreover, they are fairly easy to obtain in large quantities from the blood or urine of patients with multiple myeloma. Based on this, Kunkel and his coworkers began using myeloma cells as a tool for investigating normal antibodies. This says they're all identical. I wonder how exactly precisely they are identical they are. I mean, are they potentially folded a little bit differently? Are there very, very subtle structure variations between patients based upon the patient DNA, the donor DNA, I should say, or other epigenetic factors? I wonder. Certainly, I will agree and submit that at the time, based upon the research and I should say the equipment that they had, the methods that they had, that they believed they were all equivalent, right? Context is always important. Context is always important. The availability of myeloma cells for antibody research greatly expanded after the year 1962 when the American molecular biologist Michael Potter, again, I don't know if there's any relationship there, made the serendipitous discovery that an injection of mineral oil into the peritoneal cavity of BALBC mice, and that's a particular strain of laboratory mice, I don't know if they're humanized or not, induced the growth of myeloma cells. So these mice, which may be humanized, I don't know, when you inject them in a certain area with mineral oil, they start producing myeloma cells. And then, presumably, now you can get all of a particular type of antibody. This meant that the cells could be grown easily and indefinitely. Following this, Potter and his colleagues at the National Cancer Institute in Bethesda established a collection of myeloma cell lines for distribution to researchers around the world. Interesting. So you have a collection which suggests there were some subtle variations somehow. By the 1970s, scientists' access to myeloma cells was made much easier by the work of Kengo Horibata and A.W. Harris under the supervision of Melvin Cohn, C-O-H-N, at the Salk Institute in San Diego. They found a way of adapting Potter's mouse myeloma cells to grow in tissue culture, a technique that allows for the growth of tissues or cells outside of an organism. Essentially, tissue culture provides for the growth of cells under controlled conditions outside their natural environment. Think cloning, everybody. Cloning. The growth of such cells is helped by the use of a suitable medium, which provides nutritional elements they require. The first tissue culturing techniques date back to the year 1885 and had become a common method in laboratories by the 1940s and 1950s. Each cell type necessitated different conditions, such as temperature and various types of culture medium for optimal growth. This was perfected by a process of trial and error, as I said earlier, usually error just being factored in. The tissue culture developed for the growth of Potter's myeloma cells is described in Kengo Horibata and A.W. Harris's article in 1970 titled Mouse Myelomas and Lymphomas in Culture. Overall, the work undertaken by Horibata and Harris freed scientists from laborious processes of growing myeloma cells in mice. With the cultivation of myeloma cells in vitro, scientists now had access to a continuous supply of such cells. Cesar Milstein had obtained Potter's mouse myeloma cell line, called MOPC21, that Horibata and Harris had adapted to long-term tissue culture during the early 1970s from the LMB colleague Alan Munro, M-U-N-R-O, who had originally obtained them from the Salk Institute, where he had spent a sabbatical year. Cesar Milstein grew these cells together with George Brownlee, 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 for the purposes of harvesting mRNA as a source for studying the structure of antibodies. That's another powerful sentence. I'll reread the last two sentences here. Cesar Milstein had obtained Potter's mouse myeloma cell line, MOPC21, that Horibata and Harris had adapted to long-term tissue culture during the early 1970s from his LMB colleague, Alan Munro, who had originally obtained them from the Salk Institute, where he had spent a sabbatical year. Cesar Milstein grew these cells together with George Brownlee for the purposes of harvesting mRNA as a source for studying the structure of antibodies. That's fascinating. So, advancing the research of mRNA as a way to study the structure of antibodies. That's flipping everything upside down. And even today, what's the transfection product being given to people? The vaccines, the transfections, given to patients so that they do what? Express a protein, a spike protein, and then in turn make an antibody in response to that. That's the whole point. It's almost as if the current mRNA injections are a potentially a concealed way of studying antibody structures. And it's also a very effective way of getting around antibody patents. Holy cannoli. Everything being turned upside down. So going back up here, this cool photo here. This photo shows Michael Potter, an American molecular biologist whose myeloma cell line helped lay the foundation for the development of monoclonal antibodies. And this is him at University of Maryland. I'm not sure what the year is. And of course, Robert Gallo did all of his big work at the University of Maryland as well. This fellow here is George Brownlee. He's a British pathologist who worked with Cesar Milstein on mRNA. Based on their growth of myeloma cells, George Brownlee and Cesar Milstein were able to establish the existence of a leading sequence in the RNA of the light chain. They reported their results in a document titled A Possible Precursor of Immunoglobulin Light Chains, which was published in Nature New Biology in 1972. Now it continues to get more interesting, everybody. Let me look at the comments here. Just real quick. Thank you guys for joining. Hope that you guys are well. It's a very powerful hypothesis, Mobster. It actually bears a lot of weight because you don't have to worry about patenting what is expressed and the reaction of these chain of events from mRNA. Whereas the antibody patent issue it's a roadblock. It's a roadblock, and it also I don't know if the guest that Jonathan had on the other day, a regular guy or Sam Eagle, is there yet. I'm not saying he would agree with this speculation, but I speculate that one of the reasons why that that patent issue is so big is because by discussing it, it is revealed how little is known. Once it's revealed that there is a galactic range of antibodies and that it's largely unknown how structure correlates to different types of protection in different people, because you can have the similar structure antibody do different things in different people. There's a lot of things that are unknown. And the more that's unknown, the more people are going to go, well, that's a little bit strange. Well, and then you're going to get some people go, well, maybe the reason they're doing all this crap is because they want to figure it out with big data. So it's not going to help the bigger agenda if this is a big data mining, reverse engineering of the human genome agenda, if that's spoken about. So a nice, clever way around it is to say, well, let's just avoid monoclonals. Let's just avoid antibodies. Let's just inject the mRNA itself. And this way, we don't even need to deal with humanized lab animals. We'll just figure out what people do to them. Also noteworthy that almost all the monoclonal antibodies have lost their emergency use authorization. Also noteworthy is how some people, almost based upon political party, were religiously opposed to any deployment of monoclonal antibodies early on in the pandemic, COVID-19. And I'll throw a few more things on there. I'm going to throw a few more things on there just to get you thinking. This war that many people have seen, some people think that Dr. Michael Callahan of University of Alabama and Robert Malone are actually very close. I don't know. But they're earlier, before Operation Warp Speed was announced, they didn't seem to have a confrontational relationship. Afterwards, though, you saw funding for Inovio cut, and you saw a whole lot of money in Warp Speed go for monoclonal antibody therapies. Another part of Warp Speed that most people are overlooking. And Michael Callahan of UAB, his field of study really is the manufacture of monoclonal antibodies and using newer methods such as plant cells to make them, to produce them in greater quantities more rapidly. Not really the mRNA and DNA vaccine side. Interesting thing to note. Also noteworthy, as we've shown in the 2-minute video or 3-minute video with Drew Weissman, which I'll probably play again and maybe by the end of this video, is how he says the mRNA vaccines, and this was in 2022, had advantages over monoclonal antibodies. Now, we always heard that the reasons why the mRNA vaccines were preferred was because they could be deployed rapidly, and it was so much better than the old way of making vaccines. And what Drew Weissman slipped out, maybe he's a white hat, I don't know, but I think it was just an accident. What he slipped out, what slipped out of his mouth, I should say, was a confession that the mRNA really is about the advantage over monoclonals. It's fascinating stuff. All right, let's get back to this next section here. Fusing myeloma cells to pinpoint somatic mutation. After his letter with Sidney Brenner in Nature magazine, which had hypothesized that antibody diversity was the result of somatic mutation, Milstein began looking for a way to carry out experiments to validate the theory. This he decided to do using the MOPC21 myeloma cell line. He was joined in the venture by David Secher, S-E-C-H-E-R, I'm going to say Secher, who joined the LMB to do a doctorate in the summer of 1970. What Cesar, Milstein, and Secher wanted to determine was the rate of somatic mutation that occurred in the antibodies produced by the myeloma cell line, and to identify and characterize any variants that emerged. This, they hope, would help them unravel the process of antibody diversity as it occurred in nature. So, to review, it's known that there's a massive range of diversity of antibodies. They've detected this, And they thought that maybe this one cell line always produces the same type of antibody diversity. However, they also speculated that there would still be somatic mutation. There would still be these occasional differences, genetic mutations which are not passed on to the offspring. And they speculated that these cells would allow them to study one type of antibody and then measure the rate which it was mutating. Shortly after Milstein and Cesar began their project to investigate somatic mutation in MOPC21 myeloma cells, they were joined by Dick Cotton. Richard Cotton, come on. Dick Cotton, Cotton, a postdoctoral scientist from Australia who was interested in immunogenetics. Rapidly picking up the skills for cell culture with the help of Abraham Karpas, Karpas, who worked in a nearby laboratory, Richard Cotton soon succeeded in cloning MOPC21 cells in soft agar, AAGAR. This laid the basis for the isolation of MOPC21 mutants. And this is Cesar Milstein, obviously not a very tall guy. Or this David Cesar was just a giant. I think Milstein was just not a very tall person. With Richard Cotton's technique in place, the team then spent many months looking for somatic mutants. However, this proved frustrating. The difficulty was that such mutants rarely occur. Just 5 structural variants were isolated. For example, after 3 months of continuous culture and analysis of antibodies produced by 7,000 clones. While providing a disappointing yield of mutants, the myeloma cells nevertheless provided scientists with a window through which to see somatic mutation in cells. Importantly, mutation had occurred without any addition to the myeloma cell line. It had taken place on its own accord. The difficulty, the team found out later, was that the mutants observed in the myeloma cell line bore no resemblance to the major types of mutants found in nature. Alongside this experiment, a second project was launched, undertaken by Richard Cotton. This involved the fusion of 2 different myeloma cell lines. It was carried out using inactivated Sendai, Sendai virus obtained from Abraham Karpas. The reagent was used for the promotion of cellular fusion. One of Richard Cotton's objectives was to understand why antibody-producing cells appeared to use only one set of the parental genes to produce a functional antibody. Called allelic exclusion, ALLELIC, called allelic exclusion, this phenomenon was particularly puzzling, as in most cases, cells inherit a copy of both sets of genes from their parental cells. What scientists assumed was that in the process of antibody reproduction, one gene was silenced while the other was transferred across. By fusing 2 myeloma cell lines, Richard Cotton and Milstein wanted to see which genes could be transferred and which ones would be silenced. They were also interested in what effect such fusion would have on the structure of antibodies in terms of their variable and constant regions. In the fusion experiment, the first myeloma cell line was a variant of the mouse MOPC21 cell line that was sensitive to the chemical bromodioxyuridine. The second was a rat myeloma cell line obtained from Harvey Bazin, Bazin, in Belgium. To Richard Cotton and Cesar Milstein's surprise, the fusion of the mouse and rat myeloma cells resulted in a hybrid clone known as a hybridoma that produced antibodies carrying the genes of both parental cells. Indeed, they could find no evidence of allelic exclusion. Moreover, there appeared to be no scrambling of the variable and constant regions in the structure of the antibodies produced. Based on this, they theorized that the linkage between variable and constant regions occurred early in the process of cell differentiation before antibody production even began. Following the fusion of the rat and mouse myeloma cells, Richard Cotton, with the help of Shirley Howe, Howe, one of Cesar Milstein's laboratory technicians, continued to perfect the fusion method. One of the fusions resulted in hybridomas derived from mouse myeloma cell fusions. These yielded the same results as the earlier mouse-rat fusions. Richard Cotton and Howe's fusion efforts were greatly enhanced by the selection of a different myeloma clone known as P3X638AG8. Oh my goodness. This clone was a variant of a subclone of the P3 myeloma cell line developed by David Cesar. Cesar had made this from the cell line originating from earlier on Horibata's and Harris's adaptation of Potter's MOPC21 myeloma cell line. A key advantage of the P3X63AG8, oh my goodness, was that it was resistant to azaguanine, RAG, which is a reagent that helps promote fusion. Other myeloma cell lines were liable to be destroyed by azaguanine. And this is a popular photo here. This shows Cesar Milstein, the little guy here, and Georges Kohler, Kohler, who's a German biologist with whom he worked. This photo was taken in Kenya in 1979. Again, this is probably the most important relationship that Cesar Milstein had. In 1973, Cesar Milstein presented the results from the myeloma cellular fusion experiments conducted with Richard Cotton to the Basel Institute of Immunology. It was here that Cesar Milstein met Georges Kohler. At the time, Kohler was on a temporary secondment to the institute while completing a doctorate at the University of Freiburg. Excited by Milstein's presentation and taking an instant liking to him, Georges Kohler asked to join Cesar Milstein's research team in Cambridge as a postgraduate student. This he did in April of 1974 and soon joined in research efforts to understand somatic mutation and the mechanisms of underlying antibody diversity. By the way, if you go to Georges Kohler, Kohler, I must have misspelled it here. K-O-8. He died in 1995 at the age of 48. Only the good die young. Da, 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 da. So, finding an antibody with defined specificity. Next little section here. Prior to Georges Kohler's arrival, much of Cesar Milstein's research into somatic mutation and the diversity of antibodies had focused on a search for mutant genes within the variable region of antibodies, as this was the only portion of the antibody understood to be responsible for binding to the antigens. Yet this had proven a laborious process akin to looking for a needle in a haystack. What was needed was an antibody with a clearly defined specificity. This would provide the most effective means for detecting slight differences caused by mutations. Such an antibody, however, was not readily available. Until the mid-1970s, most of the studies Cesar Milstein and his team had undertaken on somatic mutation in antibodies were still being conducted with myeloma cells, dictated by their abundance and the fact that this was the closest scientists had come to a source of natural antibodies. However, such cells had certain limitations. The difficulty was that no one knew which specific antigens in myeloma cells antigens the myeloma cells bound to. Part of the problem was the fact that such cells are triggered by malignancy, a process that affects cells at random. Not knowing which antigen was targeted by myeloma cells was a major problem for both Cesar Milstein and others interested in understanding the molecular basis of antibody specificity. Some scientists had attempted to get around the problem by trying to induce tumors to produce antibodies to an injected antigen. These efforts, however, had come to nothing. By the time Cesar Milstein and Kohler began their collaboration, a number of scientists had begun to devise ways to make antibodies with a known specificity to a particular antigen. Discovery of natural hybrid cell producing antibodies. It gets crazier and crazier. One of the earliest was Joseph Sinkovics. Sinkovics, Sinkovics, a Hungarian immunologist based at the MD Anderson Hospital and Tumor Institute in Texas, funded by the National Cancer Institute, or the NCI. Between 1960 and 1962, while conducting research into simple viral mouse leukemia, think virus cancer program, Sinkovics happened to come across a mouse lymphoma cell with virus-like particles on its surface, a highly unusual characteristic which prompted an immune response in mice able to destroy the cell. Keen to learn more, Sinkovics and his team began looking for ways to grow the cells in tissue culture. Initially, this proved an uphill struggle, but by 1966 and 67, they had managed to grow a new cell line in suspension spinner cell cultures. This they achieved by cultivating the original cells in culture with tissue taken from the spleen of mice that had rejected such cells. Man, these guys go through a lot of mice, eh? Much to the surprise of the team, the new cells appeared to contain sets of chromosomes unlike previous lymphoma cells, which had just 2 sets of chromosomes. The new cells also grew more vigorously when injected into the adult mouse than the previous cells, forming huge tumors, which presented as abnormal fluid in their abdomens. On further investigation, the team discovered that the new cells not only produced virus particles, but also antibodies against the mouse leukemia virus. That's what Alice Moore was studying too. Further experiments with mice indicated the antibodies were highly specific against the virus. Indeed, they were better at neutralizing the mouse leukemia virus than anti-serum taken from rabbits or mice immunized with the virus. Sinkovics hypothesized that the antibodies were the result of a natural fusion of splenic plasma cell with the mouse lymphoma cell. This is crazy. Such a fusion, he argued, was highly exceptional and would only occur under special conditions. He believed it was due to, quote, a specific immune reaction between the budding virus still embedded in the cell membrane and the antibodies retained before their release on the surface of the immune spleen cells, unquote. This process generated a hybrid cell, later known as a hybridoma, capable of producing antibodies which incorporated the immortal qualities of the malignant cell. Between 1968 and 1969, Sinkovics presented his findings to several international and American conferences, during which he showed a sketch of his hypothesis. Yet, to his disappointment, the significance of what he had found seemed to bypass everyone who heard him. As he recalled, no one rose to ask a question or comment on what he had found. In spite of this, Sinkovics and his colleagues continued to work on their cells. By 1970, Sinkovics and his colleagues had shown that fused cells could grow in suspension culture for many years, and continued to produce specific mouse leukemia virus neutralizing antibodies. Furthermore, they had devised a method to purify the antibodies. That year, however, they were forced to abandon the project because the National Cancer Institute refused funding to extend the project despite finding it to be scientifically sound. Moreover, the team could not envisage any therapeutic applications for the antibodies. And continuing on, antibodies from cloned B cells. Soon after Sinkovic and his team had developed their antibodies, another group at the National Institute of Medical Research, led by Bridget Ida Asconas, Askonas, Asconas, an Australian-Canadian biochemist, hit upon a way of generating antibodies by cloning B lymphocytes, a type of white blood cell, in genetically identical irradiated mice. This they did as part of a wider project to understand the process underlying the generation of B cells and antibody diversity. Called E9, these clones produced antibodies that bound to a hapten antigen, which is a small molecule that can only stimulate an immune response when attached to a larger molecule like a protein. Labeled E9, the clones had a major drawback. They could only be maintained for a maximum of 6 weeks. Continuing on. As had happened with Sinkovic's, the generation of antibodies to a specific antigen by the Asconas team, again, the female scientist just above, would remain largely overlooked by the scientific community. More attention was paid to a technique published in 1969 by Norman Klinman, Klinman, an American immunologist based at the University of Pennsylvania, with an attachment to the Wistar Institute, Wistar. That means we definitely want to know about this guy. His method required a number of steps. First, a mouse would need to be treated with radiation to destroy its antibody-producing capability. It would then be injected with new antibody-producing cells, some of which lodged in its spleen. Oh, these poor mice. This is awful. Once this was done, the mouse's spleen was cut into cubes and individually grown as hybrid cells in tissue culture with an added antigen. If an antibody-producing cell was present within a given fragment, it would produce antibodies specific to the antigen, called monofocal antibodies. These antibodies could then be isolated from the culture medium and used for experiments. Wow. You know, this is the type of stuff, guys, where if you see your kid, if you open up your kid's bedroom door and, like a little 8-year-old kid, and you see them doing this type of stuff, you call a therapist. I mean, this is insane. You call a therapist. Like, what happened to little Jimmy? Where did we go wrong? What did he watch on TV? What was in that TV dinner he ate? What is happening to little Jimmy to make him do this to poor mice? Oh. Klinman published his technique in an article titled "Antibody with Homogeneous Antigen Binding Produced by Splenic Foci in Organ Culture" in 1969. The splenic fragment system marked a major milestone. Importantly, it showed that one cell produced only one antibody. The technique was soon adopted by a number of other scientists. One of the first to do so was Walter Gerhard, Gerhard, a Swiss-trained physician who had a postdoctoral research position in Klinman's laboratory in the early 1970s. By early 1975, Gerhard, by now at the Wistar Institute as well, had successfully generated single antibodies with known specificity against influenza viruses. The difficulty with Klinman's method, however, was that it produced only a minute quantity of antibodies. Moreover, the cells that produced the antibodies survived for a maximum of only 3 months. Boy, there's still a lot more to read here. Still a lot more to read. Boy, oh boy, oh boy. The next section is the creation of monoclonal antibodies. Ultimately, as I suggested in the first section, the first introduction to this reading, you're going to realize that one of the most important people here in this whole story is actually Hilary Koprowski. Now, we think of Hilary Koprowski as the Polish virologist, one of the 3 people who made a polio vaccine. Potentially, some of his vaccine had some contaminations. Maybe he did had some accidents involving some monkeys. I don't know. Of course, he lived to old age as well. But Hilary Koprowski at the Wistar Institute, that we found that to be particularly relevant. Remember earlier we've introduced this topic as one which is probably more important than we know because of all of the connections of intellectual property and changes in patent law, potentially even a call to destroy the patent system based upon the challenges which are coming up. And we observed that the father of the very popular now Brett Weinstein, a fellow named Les Weinstein, is one of the top arbitrators in the United States of America, was the first patent lawyer hired by the United States Department of Justice in the year he was hired in 1962. And the first thing that he worked on was the Drug Industry Antitrust Act. And we know that Les Weinstein's, one of the cases that he likes to highlight was a protein patent issue that involved Centocor, Centocor. And Centocor was founded by Hilary Koprowski. It was those sets of connections and that led us to this particular long article. As I said, once you find 2, 3, 4, and then before you know it, you have dozens of loose correlations, you know something is going to be important. So I don't know if Koprowski is Hillary in this chapter here, Hila. No, I think Koprowski is actually going to be in the next chapter, unfortunately. So we're not going to get to Koprowski today. The creation of monoclonal antibodies. On pondering how to take their investigations into the somatic mutations of antibodies further, Cesar Milstein and Georges Kohler started searching for a means to create an antibody with the desired specificity. Their starting point was Richard Cotton's myeloma cell fusions and the experiments by Gerald Schwaber, Schwaber, and Edward Cohen, Cohen, at the University of Chicago. In 1973, these men had succeeded in producing a hybrid cell line able to secrete both myeloma and lymphocyte-derived antibodies by fusing human lymphocytes with mouse myeloma cells. These earlier techniques, however, had a major limitation. Crucially, the antibody-producing cells did not survive long, and the antigens and the antibodies targeted remain unknown. Cesar Milstein and Georges Kohler wondered whether one way to resolve some of the difficulties encountered by previous researchers was to fuse a normal B cell from the spleen of a mouse immunized with a certain antigen, which itself produced an antibody with known specificity with a mouse myeloma cell. By doing this, they hoped to transfer to the antibody the trait of immortality from the myeloma cell. If it worked, they would have access to a hybrid cell or a hybridoma, not only capable of secreting antibodies with known specificity, but also of surviving indefinitely. Initially, 3 different myeloma cell lines, labeled P3, 289, and P1, were chosen as the fusion partners for the spleen cells in the experiment. After a number of trials and errors, however, Kohler determined P3, X63, AG8, the cell line used by Richard Cotton, to be the most promising myeloma fusion partner. This feels like it's really down in the weeds. Even I'm losing it a little bit here, guys. So just kick back. Richard Kohler chose to grow the fused cells in a hypoxanthine aminopterin thymidine, or an HAT medium. The medium had first been used for the fusion of tumor cells back in the 1960s. Like Richard Cotton before him, Kohler also added inactivated Sendai virus to the culture, as this was known to disrupt the membranes of cells and thereby promote the fusion process. The antigen Milstein and Kohler decided to target was sheep red blood cells, because the mouse's immune system was known to react vigorously against them. Moreover, antibodies against such cells could be easily detected by a plaque assay test, a procedure developed in 1963 that had become common in laboratories by the mid-1970s. Milstein and Kohler's fusion project was well underway by December of 1974. It was undertaken with the skilled technical assistance of Shirley Howe, Howe. Towards the end of the month, Kohler could see a number of cells growing in the medium, but he did not know if any had the desired specificity for the sheep red blood cell antigen. Before he could test the mixture with the plaque assay, however, Kohler spotted contamination of the mixture. With the cells no longer viable, Richard Kohler was forced to start the fusion process again. By the end of January 1975, he finally had some cells for testing, and he decided to perform the first plaque assay for the experiment on January 24th of 1975. This involved testing cells he had cultivated 4 days earlier through the fusion of X63 with the cells taken from a mouse immunized with sheep red blood cells. And there's a little diagram showing the steps involved in the test. Richard Kohler started the plaque assay test at 5 p.m. and returned home in the expectation that the process would take some hours before the results would materialize. A few hours later, he returned to the laboratory, bringing his wife, of course, date night, as company to see what he anticipated to be boring results. Much to his amazement, Kohler spotted green halos in the on the 2 plates on which the 2 tests had been performed. He was so happy with what he saw that he shouted and kissed his wife. That's the perfect end to any date night. The test not only showed the hybrid cells were capable of secreting antibodies to the sheep red blood cells, but they also produced large amounts. The frequency of antibodies was far greater than anything he or Cesar Milstein predicted. Hey, John G., thank you. John G. says in the chat, learning so much about the hidden war of antibody mRNA technologies. Thank you. I appreciate you being here, John. We are learning a lot of stuff together on this one. I thank you for the PayPal contribution also, John. I noticed that. I need to thank other people as well. It's all been super helpful. So thank you, John, and thank you for all of the other supporters that I do not call out. Thank you, by the way, if people have wrenches in the chat. Also, also, you do such a good job that I hardly ever have to even think about it. So thank you guys. Following this success, Kohler and Milstein repeated their experiments twice more to check the technique was reproducible. When these experiments proved positive, the two scientists realized that they possessed a tool scientists had been striving to make for many years. Critically, they had created an immortal cell line capable of producing an endless supply of identical antibodies with known specificity. The method would later be dubbed hybridoma technology, and the antibodies it produced, monoclonal antibodies. So at that moment, they named it monoclonal antibodies. Monoclonal to signify it's a single strain that's being cloned, to signify the fact that they were derived from a single hybrid cell. In May 1975, the two scientists submitted a paper announcing their experiment to Nature magazine, one of the most prestigious scientific journals. In this paper, they pointed to the fact that their technique could have major benefits for both medicine and industry. Yet the importance of their achievement was missed by the journal's editors, who asked for the article to be shortened and failed to include it in the section reserved for findings considered to be of leading significance. In the end, the article was published in Nature in August of 1975. In later years, there would be much speculation as to who originally conceived of the experiment to generate the first successful monoclonal antibodies. Kohler and Milstein responded in 1984 when they were jointly presented with the Albert Lasker Basic Medical Research Award, stating publicly, quote, both the conception and execution of the work was the result of close collaboration between us and the skilled technical assistance of Shirley Howe, Howe, unquote. Milstein would later emphasize that monoclonal antibodies had been made as a tool for answering a basic scientific question and not for any particular practical application. Interesting. So they didn't make them initially to use as a tool for production. They made them as something to understand how things were working. Last little section here, and then we'll wrap up. Wow, we're already an hour and 17 minutes in. Holy cow. A crisis and reproducibility. And there's links here to a bunch of Kohler Milstein statements and documents. I'm sure that those are quite interesting. I will probably, again, I need to download and archive this whole article still. Just at the point that Kohler and Milstein's paper was accepted for publication by Nature, the two scientists faced a major crisis. Suddenly, their technique stopped working, and they were unable to achieve any fusions. Why am I not surprised? This came as a total surprise to them, not to me, as it had worked on 7 previous occasions. Increasingly concerned as subsequent attempts failed, Milstein began to wonder whether, in fact, they should withdraw their paper from Nature. With the validity of their results threatened, the two scientists faced 6 stressful months trying to fathom what had gone wrong. Their anxiety was not helped by the very basic conditions in which they were performing their experiments. In his manuscript entitled, "In the Early Days," Cesar Milstein said, quote, "In the early days, the tissue culture laboratory was a room in the basement where the power switches of the laboratory were located. Effectively, we only had one hood and several feet of bench. The laboratory where we were working was so overcrowded, and I was so concerned about the bacteria which was being used all over the place that I preferred the situation located 4 floors away from my own laboratory. It contained the electricity switchboard, so we were not allowed to have water services inside or any other device which could mean flooding or splashing. It had no windows. The covering of the floor and the painting of the walls was totally unsuitable. My only argument in its favor was a report I read somewhere saying that in a microbiological laboratory, which had been suitably tested, the least contaminated area was found to be the lift shaft. How did my assistant Shirley Howe survive those conditions? I don't know, but I should point out that at one stage, we became somewhat concerned about her health. The what? The lift shaft? What? I feel stupid for asking this, but what's a lift shaft? Hopefully, the vertical shaft in the building which contain a lift elevator performing a cab. Wow. So there we go, everyone. Earlier I said cell culture is it's magic. It's a black art. And here they say that they had to use the elevator shaft. When they used the elevator shaft, they were able to reproduce the results. They couldn't do it in the lab. They couldn't do it in the secondary lab they didn't like. They found the elevator shaft. Earlier I said that these cell cultures are so, it's repetition. And if at some point an experiment works because the dish falls off onto the floor and someone steps in it after going in the gym, then sure enough, that will become part of the steps of producing that cell culture. And here they had to produce it in the elevator shaft. It's hilarious. So when everyone in the world is getting injected with all kinds of stuff that does all kinds of miracles, as the stupid little pictures show, just think that behind the scenes, there are some scientists, perhaps well-meaning, who are with little cell cultures all over the building, in different rooms, in the elevator shaft, and with the lights on, with the lights off. Someday you may be at Target shopping for some socks, and you may part the socks, and there is probably someone with a dish saying, you know what, this is the only place I could get this cell culture to grow, is in the sock section at Target. You never know. You never know. Not only did Milstein and his team have to work the very basic, they had to work in the elevator shaft, work in the basic and hazardous conditions. Oh, this is beautiful. This is beautiful. Yeah, they believed that the elevator shaft had the best control of contamination. No, because you know why? No, maybe no one else was there. It was probably a combination of the light, the temperature. Maybe they didn't know. It's just, this is the magic involved. It's not that it doesn't have an impact. The question, it highlights how much is unknown, how much is luck. They shouldn't be embarrassed about it. There's nothing embarrassing about it. It's kind of fascinating, in fact. However, I bring it up because our friends and our family, our world is being vastly oversold about what is known and what is possible right now. That's the takeaway from this. It's not that there's anything necessarily wrong with this. It's just that we are being oversold as to the state of the art of technology, of the capabilities of things. Just much is still unknown. And I'm okay with that. Anyways, I think the next section. So the next section, which we're going to be our next reading, is sure enough, when it comes to antibodies. I didn't make this up. This is a 10-year-old article. The person that wrote this article dedicated a whole massive chapter on patents. Do you think they did it because they wanted to bore people to death? I don't think so. I think because the role that patents have played with how companies have been able to succeed with respect to how people have been potentially available for medical experimentation, with respect to which political movements have access to what, the role that patents play is enormous. Someone else who probably spent 6 to 12 months writing this article, researching this article, decided that the patent issue was that important. And by the way, thank you, by the way, Marie. God bless you. Thank you, thank you, thank you. You are amazing. You're amazing, too, for being here. That was not exactly a captivating article at every twist and turn in every paragraph. So thank you for still being here. What brought us here, ladies and gentlemen, boys and girls, what brought us here was Les Weinstein. What brought us here was, why is Brett Weinstein and his brother such a big name? What brought us here was the antibody patent paradox. What brought us here was Hillary Kaprowski, Inovio. That's what brought us here. And sure enough, a huge part of the antibody story, and the antibody story, the monoclonal antibody story is also itself defined by using mRNA to study antibodies. And antibodies, again, being proteins, it's really protein patents overall. And protein patents is really patenting life overall. So this is why we did that chapter, and I have to keep reminding you, and even reminding myself, why the heck are we reading this? Who cares about patent? Who really cares if this company or that company makes a few extra bucks? Investors do, but so what? How could the patent issue really be so big that it's driving policy decisions, which are impacting our freedoms, which are impacting the implementation of our Constitution within the United States? Well, here we go. So with that said, everybody, next chapter will be good. I did say I was going to list out all the names in our previous chapter. We're already an hour and 27 minutes in. That's been a long time. I will do it, though. I will list out the names. I should create pages for each of these individuals. I will do my best with the time that we have. Patent Saga is going to be a great chapter. Thank you, John. Thank you, Marie, for your contributions. I appreciate it very much. Thank you, everyone, for being here. God bless. Have a great rest of the day. Stay safe. Keep learning. Keep curious, but not so curious that the thing that's between you and your loved one and your families is a screen. Put the freaking screen down for a little while. Spend some time with them. And that's all, everyone. Take care, everyone. God bless.